There we go. Okay, let's go. So the next section is the principles of adaptive immunity. So this is going to be all the stuff that makes T cells tick and B cells tick and kind of the way they work. And we will have individual chapters on each of these things that are going to be pretty intense, pretty in-depth, but they're going to teach you immunology at the level at which it actually works, which is kind of the molecular level. Okay, so what's next? Well, next thing that has to happen is antigens, which is a little piece of that virus or bacterium or whatever is making trouble, are going to be presented on antigen receptors, and these are going to cause white blood cells to acquire effector activity. So that means they're going to make white blood cells change in a way that makes them effective against that virus or bacterium or whatever. Okay, so how does this work? Well, a couple different ways, yeah. So two of the main ways are that things related to B cells are going to make antibodies. And an antibody is a molecule that's Y-shaped like this. It's actually two of these green chains and two of these yellow chains, and they are kind of stapled together with what are called disulfide bonds. So you can see two down here, one up there, one up there. I think this is an IgG type 1 that we're looking at. Depending on whether you have an IgG type 2, A or 2B or 3, or like an IgM or an IgD, IgE, or an IgA, you will have different patterns of disulfide formation. And if I say Ig anything, it stands for immunoglobulin, which is the fancy immunology word for an antibody. So IgA, you could also say, is immunoglobulin A, or an antibody, an IgA type antibody. There we go. Shown down here at the bottom is the T cell receptor. So this is what T cells are trying to make, while B cells are trying to make one of these. And the T cell receptor is kind of like just the top part of the antibody. It has an alpha chain and a beta chain, or there's a different kind of T cell that has a gamma chain and a delta chain. And it's going to try to find some way to mess around with this top bit to the point that it actually binds to whatever it is that's attacking your body at the moment. And that's exactly what happens here. So there will be a library of top bits, middle bits, and bottom bits for antibodies and T cells. And there are potentially millions of ways. So in a normal human genome, there are about 60 million combinations, between 30 million and 60 million, depending. Some of these genes you get more copies of or fewer copies of, depending on just your genetic luck. But a lot of different copies, a lot of different possible base versions of antibodies and T cells. And then beyond that, we actually go in and our bodies will put in random mutations, particularly in this top part, to try and just search for a better way to stick to a protein. So these proteins make a shape, and that shape's job is to latch onto a little piece of a bacterium or a virus or something like that. And that is the way that the adaptive immune system works, by making one of these antibodies or one of these T cell receptors. Those are the big ways. So compare the way that they recognize antigens. Remember, antigens are the bad thing that we're trying to stop. So you can have different antibodies that will recognize different parts on the surface of even the same protein. That's fine. And they will be able to bind it and latch onto it. And so the shape of this antibody and the distribution of positive and negative charge, or hydrophobic and hydrophilic atmospheres, if you like, will find a match basically for whatever the epitope is. So there is a way to stick to any piece of protein. And generally, antibodies are going to bind to proteins, although you can make antibodies against sugar or lipids or different molecules or even some heavy metal atoms. You can make an antibody against a cadmium atom, of all things. Okay, now T cells work in a different way. So this is the way the T cells work. Antibodies just stick to stuff and then wait for another white blood cell to come along and pick up the thing that the antibody is stuck to and destroy it. T cells will take a little piece of whatever protein they've picked up or a protein that they are making at the moment, shred it into little tiny pieces. A small fraction of those pieces, maybe 1%, maybe less, can actually bind to one of their molecules. And so this is an MHC molecule here. And remember, MHC is the way that cells show a little bit of whatever they're making at the moment. And so they're going to bind it, and it actually does bind in a groove right here on the top. The whole thing kind of looks like a clothespin, maybe. The old kind with a spring in the middle that you can open and shut, if that paints a picture for you. And then the T cell receptor will come along, and it will bind to a protein, which is in the middle, that is stuck to an MHC molecule. All right. Once each of these cells gets activated, it can start to divide. And so an activated cell will divide, or a progenitor can divide and make lots of different activated cells. Activated cells can divide and make more copies of themselves. This is called proliferation. And the cells that bind to foreign antigens are the ones that get to proliferate. So if you think what a cell is making in a healthy person, it's going to be making all your regular proteins. You shred some of those regular proteins. You present them on your MHC, major histocompatibility complex, and you show them to the immune system. And if your T cells were to latch onto that and recognize it as foreign, you would have an autoimmune condition. Something like irritable bowel syndrome or Crohn's disease or systemic lupus. That's another one. But there are a lot of these things. And but if you can make a response to a foreign antigen, then this is rewarded. So there is a system built in where if you're binding a self-antigen, any cells that bind those could be and actually should be killed off, just murdered by other parts of your immune system. They're basically told, you bound the wrong thing, you need to kill yourself. And, we'll let another T cell try, but you have made the wrong choice. If you bind a foreign antigen, there's no cell there to say that this is foreign, and you get signals that say, well done. And we'll go over all of these signals, but that just leads to more production of those cells that happen to make the winning combination, and that then go out and try to mop up all of whatever it was. And so you'll have many different kinds of antibody and many different kinds of T cells that will be involved in the response to a single infection. Because each of those T cells or antibodies is only seeing a little tiny part of the virus or bacterium at the same time. And so together they can fight different parts of it, and they can basically gang up on whatever it is that's attacking you and making you sick. Yeah. All right, so that's what we just talked about. And then where do these lymphocytes, white blood cells, go? So if we say lymphocytes, you're going to assume white blood cells, things like T cells and B cells in particular. And there are actually a lot of places in the body where these things hang out. So the spleen is the biggest place. That's like, whatever the Dallas AT&T Stadium is, just, with a giant jumbotron. That's where every cell in the every cell in the immune system, rather, wants to be. Or the Superdome if you're a Saints fan for some reason, something like that. But there are also going to be like tonsils. There are also going to be lymph nodes throughout your body. There are going to be little patches all around the small intestine, these Peyer's patches. And all of these are going to be places where a different population of white blood cells is going to be hanging out. There will be some of them that hang out in your bone marrow. And so there's a chance, no matter where a antigen is recognized, that you're going to bump into the right B cell or T cell. And so antigen-presenting cells are going to go along, and they're going to run along the lymphatic system and bounce from one of these to another until they find some kind of white blood cell that's willing to take them seriously, basically. Okay, and so here is one of our dendritic cells. So this one has picked up something bad. It's doing macropinocytosis, which is a way that it can kind of wrap its arms around kind of a big thing, like a whole bacterium, or it can be a whole cell even, one of your cells, and then engulf it and pull it in and digest it. And so then these dendritic cells, once they've done that, if they do that and they get some other signal that sort of tingles one of their pattern recognition receptors, which are those things like the toll-like receptors that we mentioned earlier, then they will be activated and they will run for the nearest lymph node, and they'll go and bump into a bunch of T cells or B cells, and they will try to find some that are able to recognize it. And then they can go, or they can go into a different one. They can potentially move from one lymph node to another if they have to, until they find the right match. And this is going to be going on all over the body. You'll have different dendritic cells reporting to different nodes. It's like, information coming into all these little tiny police precincts. They're like, oh, there's a bad thing happening over here. Like an emergency response center that's correlating all these little tiny emergency response centers. Each one sends out its own little immune force to try and deal with the problem. And so dendritic cells are kind of a bridge, yeah. They are taking whatever it is that these guys, neutrophils, eosinophils, basophils, and monocytes, whatever they are responding to, the dendritic cell will pick up some of that and bring it over here and show it directly to a T cell or a B cell. Or it'll show it to a helper T cell, which will then kind of help and go and stimulate the right T cells and B cells. And yeah, so dendritic cells are kind of a bridge. And macrophages can be a little bit bridgy. They can do a bit of that as well. Neutrophils can do a tiny bit of bridging, but mostly they just eat to destroy. Yeah, but dendritic cells eat to go and report to the adaptive immune system. How do they do this? Bloodstream. Bloodstream is everywhere, as are the lymphatic vessels, which are like a version of the bloodstream. They run parallel to it, but you don't have any actual red blood cells running around in there. You just have white blood cells in the lymphatics. So they can be a little smaller, but they get pumped around by the same force. They feed into the heart and get fed back out from the heart. So even within a lymph node, a little tiny lymph node, you'll have all kinds of zones. And in each zone, you'll have different cells that group up. And so you'll have a zone with mostly B cells, a zone with mostly macrophages, and plasma cells are B cells that have learned how to make an antibody, so they're like mature B cells. You'll have sort of sorting areas in the center. You'll have areas where you find T cells. So the pericortical area is going to be mostly T cells in here. And each of these zones represents a place where a dendritic cell can potentially go to show off what it's found and to generate a specific kind of immune response. And so various signals that maybe make one of these zones a little more or less appealing to our dendritic cells are going to influence the kind of response that you make. Because if all of the dendritic cells report to the B cell zone, then you are going to make a great immune response in terms of antibodies, but you may not make a T cell response. You may not make any other kind of immune response. And in some cases that'll be good, and in some cases that'll be real bad. So it's just, it's a thing that is carefully regulated and can be controlled. The spleen is the same deal. It's got all kinds of, if you ever see a spleen cut open, it's got all kinds of these little zones with different cells that you can actually see. So here's our little, our little germinal center, little baby cells, and you've got your marginal zone, you got your B cell zones, and yeah, each zone is going to have a different group of similar cells located in it. And they start out very well organized before an infection. And what you'll notice is that if you get an infection and the infection hits some of these white blood cells, you will get just total dysregulation. It's going to look like chaos in a spleen or in a lymph node because it's actually infected. So yeah, one thing that infections do is they sometimes will disrupt our little lymphoid organs, which is a nasty thing to do now, isn't it? So this is going to be one of those little patches, which is going to have M cells and something that's kind of like a dendritic cell. And these guys are going to be located along the intestine, and they're going to pick up little bits of whatever it is that you eat. and they are also going to report that. And so this is going to be how you fight infections that are down your gut. But also, if you eat something, this can end up, and you eat something for long enough in like large enough amounts, this can end up causing a thing called tolerization, where your immune cells learn to not pay attention to this thing. So one of the treatments for things like peanut allergies is actually under doctor's supervision, with like an EpiPen at the ready, to eat small quantities of peanut protein. And you sort of build this up over time. This is the way that sort of immune therapy for certain allergies works. They feed you little bits of that thing and allow these patches in your intestine to start to make the right immune response. Okay, and we're getting to the end of this section, but yeah, there will be, you can break the immune response down into different components, like a primary response. So the lag phase is going to be when you're trying to make your T cell or B cell response, and you'll have a second lag phase up here. You may have a response to one part of the antigen or one particular antigen that takes place, and then making a response to a second antigen from the same thing can lead to the signals that cause you to actually make a big response to the first antigen again. And so as you get more of these white blood cells that are learning how to recognize whatever it is that's making you sick, you'll find that they can stimulate each other. So this is why we sometimes have more than one dose of a vaccine. The first time you might make this response, but the second time you might see something else. Just, your immune system happens to notice something else. And so you'll get all of the old response, which will be a little bigger than it was last time, and a new response on top of that. And you can potentially keep on boosting this over and over again. Sometimes they'll do 3 or 4 boosts to get you up to a high enough level of immunity that it's actually going to last. Because once your immunity goes up, eventually it's going to start to come back down again. And the higher you can push it with a vaccine or during an immune response, the longer it's going to be around in general. Okay, so that's the end of that section of this particular